Hello, this is Purva Thakur. I'm third year student from Group 10A. I'm going to tell you about the introduction and the pathogenesis of HIV. HIV is the causative agent of AIDS, belongs to a family that is Retroviridae. It is a spherical and an envelope-shaped structure which is 90 to 120 mm in size. It has a genome that is a single-stranded RNA and it has an enzyme that is a reverse transcriptase. Basically, HIV virus is transmitted through the unprotected sex, from the blood transfusion, and from the mother to fetus by the placental route. Pathogenesis of HIV. HIV virus has the GP120 protein on its surface which attaches to the cells which have the CD4 molecules over their surface. Macrophages, dendrites and T cells have these CD4 molecules on their surface which helps to attach the virus on their surfaces. After that, the uncoating happens which results in the entry of the viral materials from the viral part to the cells. After this, the reverse transcription takes place which converts the single-stranded DNA into the double-stranded, goes inside to the nucleus where the integration and transcription happens. After the transcription is completed, the mRNA comes out from the nucleus, results in a further process that is a translation. After the translation, the assembly happens, and when the assembly is completed, the viral is ready and it's buds off and further process to the maturation. And after the maturation process, the viral multiply itself into the number of copies as you can see there. These number of copies infects our CD4 cells and destroy them, which makes the person immunosuppressant. As you can see, the guy is sad because he becomes immunodeficient. That increases his risk of getting the infections and the tumors very easily. Thank you for your attention.
Hello, I am Monica from Group 10A. I am going to tell about serological test for HIV. A serological test is used to detect the presence of antibodies against HIV in a person's body. The HIV serological tests are commonly used to diagnose and monitor HIV infection. The two most common serological tests for HIV are Western blot. This is a confirmatory test that is used to done after a positive ELISA test. It detects the specific antibodies to different proteins of HIV in the blood. And next one is enzyme-linked immunosorbent assay, ELISA test. The enzyme-linked immunosorbent assay or ELISA is an extremely sensitive test that is used to detect antibodies or a specific antigens. The direct ELISA is a test for the presence of antigens. In this procedure, unknown antibodies is observed to the inside of the well in a microtiter plate. After rinsing to remove excess antibody, the sample suspected of containing the antigen is added. Next, an enzyme-linked antibody that can react with the antigen is added. If antigen is present in the well, the enzyme-linked antibody binds to it and is retained the colorless substrate. For the enzyme is then added development of color indicate the presence of the antigen. In the indirect ELISA, antigen is added to the microtiter plate well and the antigen attached to the wall of the microtiter plate. After rinsing to remove excess antigen, the serum suspected of containing the antibodies is added. Enzyme-linked antibody capable of reacting with the constant region of other antibodies is then added after followed by addition of the colorless substrate. Development of color indicate the presence of the antibody being identified.
Hello, my name is Vishal. Today I will explain the prevention of HIV and prophylaxis. Prevention. Use condom every time when you have sex. Limited number of sexual partners. Get tested and treated for STD. Do not inject drugs. Prophylaxis. Prophylaxis means to prevent or control the spread of an infection or disease. That means taking HIV medicine within 72 hours after the exposure to HIV to prevent HIV infection.
Hello everyone, myself Sunny. Today I am going to explain you some important strategies for the HIV testing. As we all know that HIV testing is essential part for the HIV prevention, treatment and care. Here are some important strategies for HIV testing. The main strategy is routine testing. HIV testing should be a routine part of healthcare, mainly for the people who are at higher risk of infection. The community CDC recommends that the people between the age of 13 years to 64 years be tested at least once as a part of healthcare, and also for the pregnant women. Other strategies are like target testing, community-based testing, self-testing, partner notification, and rapid testing. In rapid testing, we can get the result within the minutes, which mainly help the individuals to take the appropriate care and treatment quickly. It is important to note that HIV testing is voluntary and accompanied by the counseling before or after the test.
HIV pathogenicity. HIV infects white blood cells of your immune system called CD4 cells or helper T cells. It destroys CD4 cells, causing your white blood cells count to drop. HIV innately makes you feel sick with flu-like symptoms. Then it can hide in your body for a long time without causing noticeable symptoms. AIDS can cause rapid weight loss, stomach redness, mouth or genital ulcer, fever, height, sweats, and skin discoloration, and cause additional symptoms. Thank you.
Myself, Kirwadan Singh. I am going to tell you about the clinical features of HIV and AIDS. The primary infection signs and symptoms are muscle aches, chills, mouth ulcers, fever, sore throat, fatigue, night sweats, swollen lymph nodes, and skin rashes. I'm going to tell you about secondary stage of symptomatic infection. Pneumonia. Individuals infected with an immunodeficiency virus HIV are more susceptible to bacterial infections because of defects in both cellular and humoral immunity. Streptococcus pneumoniae and Haemophilus influenzae are the most common cause of bacterial pneumonia in HIV-infected patients. Swollen lymph nodes. Swollen lymph nodes are central to the pathology of HIV. They contain large population of HIV-targeted CD4 plus T cells and other immune cells organized in specific regions of the tissue to facilitate immune response. Here you can see the woman is suffering from swollen lymph nodes. Shingles. A person is more likely to develop shingles if their immune system is compromised, and this can include people with untreated HIV or stage 3 HIV. People with CD4 cells and higher HIV viral loads are more likely to develop shingles and have several complications.